Hi there. I'm going to be doing a review video for you for quiz number one. In class, I already gave you a list of topics that you need to know for quiz. If you didn't receive a copy, you can also find it on eCentennial. So let's go through these things one by one. Okay, so first thing you need to know for the quiz is the characteristics of living things. There are 7 characteristics of living things that we talked about. So I'm just going to go through each one of them with you here. So the first one is complexity. Complexity and organization. Complexity and organization, meaning that living things has order to it. So symmetry, for example, there's left-right symmetry, and we look at how like a seashell, there's like certain geometry associated with it. So that tend to be a characteristic of living things. The next one is the ability to maintain homeostasis. Homeostasis means an internal balance. Internal balance. Despite of changes that are happening, in the surroundings, you are able to keep the inside relatively stable. For example, keeping a body temperature 37 degrees Celsius, maintaining a blood pH of about 7.35 to 7.45. Um, these are certain set points, certain numbers that we need to maintain in order for the body to be compatible with life, and the ability to keep those um, uh, numbers. That's what we mean by homeostasis. Number 3, I'm just listing it as they are coming to my mind, so they might not be in the same order as the PowerPoint slides, but they're all there. Right? Growth and development. Organisms tend to grow throughout their lifetime, and sometimes they develop into something very different, like the caterpillar into a butterfly. We also have the response to stimuli. Okay, so if there are changes in the environment, your body is able to react accordingly. If you touch something hot, your hand's going to recoil. That's a response to an external stimulus. In order to grow and to develop, we need to have energy. So there is a requirement for energy. How the organism acquire the energy might be different. Plants are able to make their own energy using the sun, whereas for animals, we need to consume other organisms to make food, to make energy. So the mode of energy acquisition might be different, but the need for energy remains the same. Number 6, reproduction. Talk about how living things are able to reproduce more of their own kind. And last but not least, adapt and evolve. So the entire population will change over time so that it will be more adapted to the environment that it's in. So those are the 7 characteristics of living things. We talked about in order to maintain homeostasis, there are different ways, different mechanisms that your body is going to do it for you, or there are things that you can do yourself to maintain homeostasis. So let's say it is very hot outside, okay, very hot outside. So physiologically, what does your body do for you to cool you down? Well, you can sweat, right? That's going to cool you down. Your blood vessels, if you look at the blood vessels on your arm on a very hot day, they tend to be more prominent. They tend to be closer to the surface. So blood vessels dilate and help basically vent off some of the heat that are trapped in your body. So together, these things will cool you down. In terms of behavior, things that you can do to cool yourself off, you can wear something thinner. Wear something thinner, so put away the jacket, right? Put on a t-shirt or something. You can drink something cold. Okay, or you can like turn on the fan or turn on the AC, right? So these are different ways that you can cool yourself off, things that you actually do yourselves as opposed to your body doing it for you. That's the biggest difference between physiological and behavioral mechanisms. Okay. Next, we talked about quantitative and qualitative observations. Quantitative, the one with the T, that is using measurements. Measurements. Right? So let's say you have an apple. Okay, you have an apple, and you want to describe this apple using numbers, using measurements. So you can say, oh, it is like 700 grams. That's how heavy it is. That's a number. That's quantitative. You can say that the apple is about, 5.2 centimeter wide. Okay. Anytime you're using numbers, anytime you see number in the description, it's going to be quantitative. Qualitative is using our senses. Okay, so what you see, how it smells, so on and so forth. Back to our apple example, we can describe it as red. That's what we see. It is shiny, for example. Shiny. It is sweet if you eat it, or sour maybe. And it feels smooth when you touch the skin of the apple, right? So all these things are just based on your senses, right? And the biggest problem with qualitative observation is that they are personal, right? They are subjective. It depends on your perspective, right? What's sweet for one person might not be sweet for another. That's why, generally speaking, quantitative is a little bit more objective, a little bit more appropriate for some kind of description. All right, inductive and deductive reasoning. I know in class we had a little bit of trouble, learning about that. And don't worry too much. There's only one question on the quiz about inductive and deductive reasoning. So for example, we can say, Ebola is a virus and it requires a host to replicate. Okay, and then we can pick another virus. Like HPV is a virus and it requires a host to replicate. We can say influenza is a virus and it requires a host to replicate, and then maybe just one more. Okay, we can have like I don't know what other viruses are there, like hepatitis virus, right? Or yeah, hepatitis virus. Yeah, it is a virus, okay, and therefore requires a host to replicate. So all these individual observations, right? We can conclude, we can come up with a generalization and say that all viruses require a host to replicate. Okay? So this is going from individual observations, specific observations, to generalization. Okay? This direction of reasoning is called inductive reasoning. Okay. So over here we can say, okay, now there is a new virus. Okay, I mean it's not new anymore. It's like 5 years ago. But then let's say, COVID-19 is a virus. Okay? And then based on our generalization, we can say it requires requires a host to replicate. So that here, applying the general statement to a specific case, that would be deductive reasoning. Okay. So let's say you know, I say chicken pox, right? Chicken pox, right? It's caused by something called HSV. And even if you didn't learn about chickenpox at all, and I say, you know, it's caused by a virus, then based on this generalization, you can say, oh, it must require a host to replicate, right? So applying a general statement to a situation, individual situation, that's deductive reasoning. Okay. I mean, feel free to pause the video, right? Like if you need to copy some of these things down or whatever, right? Moving on, we have the dependent and independent variable. Now, we did a lot of different a case study in class. You know, we talk about the plants, we talk about you know, the cows and the milk yield. But there was a example in the PowerPoint. I'm just trying to look for it right now. That we didn't have time to go into too much detail in class, and I'm just trying to find that right now. It was the one with the microwave water, right? There you go. So the internet says if you water your plant with microwaved water, it's gonna kill the plant, right? So how can we go ahead and test this? Well, what we can do is we can actually have a bunch of plants here. Okay, so we're gonna have the little plant here, right, with the leaves and whatnot. Okay, and you know, for it to be a good experiment, you don't want to have just like a single plant in your experiment. You want to have, you know, a bunch of them, right? So I'm just gonna put a bunch of them here. Okay, let's copy 4 at a time. There we go. Okay, so let's say I have a group here, right? And I am going to have a second group right here. Okay, the magic of copy and paste. So this first group here, we're gonna say this is the control group. Control group. Control group means they receive no treatment. No treatment. And no special treatment. You just treat them like you normally would. And again, the purpose of a control group is to provide a reference or baseline to compare. Running out of space here. Give me a second. There we go. Okay. Is to provide a reference or a baseline to compare the experimental group with. Okay, so this is here my control group, and what are they gonna be receiving? They're just gonna be receiving, you know, purified water, as it says in the picture here, purified water. Okay, or normal water, right? Doesn't really matter. But the point is you're giving them like non-microwave water. And then over here, we're gonna have our experimental group. Experimental group. Okay, also known as the treated group. This is the group that's gonna be receiving the modification. So they will be watered with microwaved water. And again, just to reiterate, these are like water that has been microwaved, but then cooled back down to room temperature before you water them, right? So we have two groups here. Right? Now, what is the independent variable? Remember, the independent variable is the one that you are in control of. It is the one that you are changing between your control group and your experimental group. And in this case, it is the type of water used to water the plants. Okay, so that's the only thing you're changing. And what are you trying to measure? What are you trying to observe? Right, that would be the dependent variable. Okay, which would be like the growth of the plant. Okay, like is it gonna live or is it gonna die? Right? We can say instead of the growth of the plant, or like the survival, right? Survival of the plant. Okay, is it gonna live? Is it gonna die? What's going on? Right? So that's the dependent variable. Now, other than the type of the water, everything between the control group and the experimental group must be kept the same, right? So here is a list of standardized variables. These are things that needs to be kept the same. You can look at your notes to see what the definition of standardized variable is, but very briefly, these are a list of things that needs to be identical between the control group and the experimental group. And the purpose of having standardized variable is so that when you do see a difference in between the groups, you know the difference is caused by the change in the independent variable. Meaning, if I do see that the experimental group plants, they all die, but not the control group, I know it was because of the microwave water and not anything else, right? So standardized variable in this case would be, pretty much the stuff that you guys told me in class, right? Type of soil, temperature, right? The environment that you leave these plants in, right? Type of plants, right? Initial condition of the plant, so on and so forth. Okay? And then you can do this experiment for like 7 days or something, 9 days here it says, right? And then compare the results, right? So it turns out this is a completely false, okay? Microwave the water and watering your plant with it is perfectly fine as long as you don't do it while it's like boiling hot, right? Okay, cool it back down and do it. There's no difference chemically. There's no difference between the regular water and the microwave water. Okay, so this is as of many things in the internet, this is not true. Okay, then we talked about the Pasteur and Raydi's experiment. Like I said in class, you are not gonna have to explain them, but you definitely need to have a good understanding of what it is. For example, here is the Pasteur and Raydi. This one is the Raydi's experiment. Okay, that's like the piece of meat guy with the flies. Okay, Francesco Raydi. So things that you should know are like the dependent variable, which would be the presence of flies and maggots. The independent variable would be the type of jar that you're using. Okay, so again, all these things we did in class, you just gotta find the class notes and go through it yourself. Same thing with Pasteur's experiment. Pasteur experiment is the one with the flask, right? Remember, there are two kinds of flask. One that is like normal top, the other had the swan neck. So the independent variable would be the type of flask being used, and the dependent variable is going to be the presence of microorganisms in the broth, right? So make sure you understand how these experiments are set up. And they were both trying to test the theory of spontaneous generation, right? Okay. And it turns out that both of the experiments together, they rejected the idea of spontaneous generation. Okay, we talked about control group and treated group already in the microwave water experiment above. We also talk about placebo. Okay, placebo is it's a sham treatment sham treatment, it does not have any effects on the dependent variable. And we need a placebo when we deal with human subjects, right? Yeah. So a placebo is needed to minimize bias and psychological effect in human subjects. We can say in experiments involving human subjects. Okay. We talk about standardized variable already, again, in the microwave water experiment above. And then there is the concept of hypothesis. Yeah, hypothesis is like a tentative answer. Tentative answer to the questions. And in order for it to be valid, it must be testable. That means you need to be able to conduct an experiment to test it out. And it has to be potentially wrong, or we say falsifiable. Okay, falsifiable, potentially wrong. Okay. So that is pretty much what you have to know from lecture number 1. Moving on, lecture number 2. We talked about a lot of the chemistry concepts, but a lot of those are just like background information to help you understand the macromolecules, right? So some of the more important things that you should know for the quiz would be the elements of life. So there's carbon, hydrogen, oxygen, nitrogen, and phosphorus. These things makes up, over 96% of the human body. In addition to these key elements, there are also trace elements, right? So trace elements are elements that are required in small quantity in the body. So things like calcium, zinc, iodine, right? We mentioned about iron in class as well. So these things, we don't need a lot of them. In fact, if you have too much, it can lead to like overdose in some cases, like iron overload. That's not a good thing, okay? But if you don't have enough, that's also a problem. Right? If you don't have enough iron, you might become anemic. If you don't have enough calcium, you might get osteoporosis later on in life, right? So it's all about having the right amount of these things in our diets, right? So that we can maintain normal body functions. We talk about the concept of pH. pH is a measurement of, whether something is acidic, neutral, or basic. Okay? So if you have a solution and you want to know whether it's acidic, neutral, or basic, you check the pH. You can check the pH with, there are instruments to do that. There are some test papers that we can use to test the pH. So the pH, if it's less than 7, and it's acidic. Things that are acidic tends to be like sour, right? They're going to be corrosive. And example of acid would be things like vinegar, lemon juice, orange juice, and stomach acid, right? Those are some examples that we did in class. If the pH is greater than 7, then it's basic. Basic stuffs tend to be bitter. They are slippery to the touch, and they actually are also corrosive. And example of base would be things like baking soda, soap, right? Bleach, all these things are basic. Cleaning products are basic. Tums, right? If you have a heartburn, then you need to take some Tums and they neutralize each other, right? Neutral would be when pH is 7, and that is like pure water. Okay. Talking about water, we spent quite a bit of time uh, describing the properties of water molecules. You should know that a water molecule is made up of oxygen as well as hydrogen. And what's really interesting about water molecule is that when you have a bunch of them next to, when you put a bunch of them next to each other, I should say, there's going to be attraction between them. And the reason why they have attraction is because when you form the water molecule is polar, meaning there is uneven distribution of electron between the oxygen and the hydrogen. So the oxygen is slightly negative, and the hydrogen is slightly positive. So when you put them next to each other, the slightly positive hydrogen will be attracted to the slightly negative oxygen. And that is how you get these interactions. Okay? So these attractions between the molecules, we call them hydrogen bonds. Hydrogen bonds. And that makes the water molecule polar. The uneven distribution of electron makes the water molecule polar, and they have attraction between each other. All right. We talk about how macromolecules, the carbohydrates, proteins, nucleic acids, and lipids, they are polymers, really big, long chains of molecules that are made up of individual monomers. So let's say we have these individual circles here. They represent the monomers. Okay, and then we're going to connect them together, right? So let's see how many we have here. 1, 2, 3, 4, 5, 6, 7, and 8. Okay, so we can join them together. So watch this. Okay, number 1 connected to number 2. Okay, and every time we connect them together, we do what's called like a dehydration synthesis, right? Okay, or condensation reaction, which means we remove a water molecule. So when I connect 1 and 2, we're going to remove a water molecule. When I add on number 3, that's going to be another water molecule. When I add on number 4, 5, 6, and 7, every time we connect an extra monomer, we're going to have to remove a water molecule. Okay, so this is important, huh? When you have 8 monomers that you need to connect together, you will remove a total of 7 water molecules because there are 7 bonds here, right? 1, 2, 3, 4, 5, 6, 7. 7 bonds are needed to connect 8 monomers together. So in total, you have 7 H2O that are removed. If you want to break the polymer back into the monomer, then we're going to add water, and that is called hydrolysis. Hydrolysis. You're going to add individual water molecules between the circles, right? And then you're going to break the bond, and you're going to be able to release the monomers back. Okay. We talk about different kinds of saccharides for carbohydrates. So, for example, we have glucose, we have fructose, and we have galactose. Right, so these are monosaccharides, single sugar. And of course, we can connect them together. We can take 2 glucose and put them together. So glucose plus glucose is going to give us maltose. We can also take glucose plus fructose, and we would get sucrose. And we can also take like glucose plus galactose, and that's going to give us lactose. Okay, so these here, these are like, you know, double sugar, right? So we call them disaccharides. Sorry, I'm just gonna move the box here. Looks nicer. Disaccharide. Double sugar, right? And we can also keep on building it. Right? So in class, we talk about things like starch. Okay? We talk about cellulose, chitin, and glycogen, right? Glycogen. Let me just put glycogen here at the top. Okay. So glycogen and starch, these are storage form of energy. Storage form of energy. Right? Glycogen is storage form of energy in animals. Starch is storage form of energy in plants. Okay? Glycogen, you store it in the muscle and the liver of animals, and starch you usually store it in the roots and maybe the stem of the plants. Cellulose, this is plant fibers. Okay, like the crunchy parts of the plant, the tree bark, they have cellulose in it. Chitin is the material that makes up the exoskeleton of insects and crustaceans, right? Like lobster, crabs, that kind of stuff. Okay, so these are for structural support. These are for storage form of energy. And what's important is that these four things are examples of polysaccharide. Okay, as opposed to double, these are like really, really long chains. All right. Now, I kind of forgot to mention this one, but you know, in the dehydration synthesis here, let's take a closer look. Okay, so you know, by now you should know glucose has like a hexagon shape. Okay, you're not gonna have to draw them, but you know, I'm just gonna use hexagon to represent the glucose, and it has an OH in it. Okay, so let's say we have glucose here, and the fructose has a pentagon shape in its structure. So let's say there's like an OH here as well. So if you put them together and you join them through dehydration, synthesis, all right, what are you going to get? Well, we're going to have to remove OH from one of them, and it doesn't matter which one you take, okay? So you take OH from one of them, and then you take an H from the other one, okay? So the H and the OH, the highlighted part, it's going to come together to form the water, and then you're going to have the hexagon and the left behind O, and then it's going to connect to the pentagon. Okay? That is basically the process of dehydration synthesis. Okay, just give me a second here. I am just going to take a picture from the PowerPoint slide to show you that you actually learned this. Okay, so over here, like I'm just taking away all these like fancy stuff. Okay, I'm just showing you, okay, hexagon right here with the OH, right? And then we have the pentagon here, right, with the OH. So afterwards, you have your hexagon, the O, and then the pentagon. Right? Like again, I'm just like ignoring all those fancy stuff on the side that you don't have to know at this point. But that's basically what the reaction is trying to show you, right? Okay, that is that. Let me see what else you have to know. Okay, next we have to know the components of the different types of lipids and the function and the structure of the different types of lipids. So we've only learned several types of lipids in class. Let me go through that with you quickly. I mean, it's in your study guide as well, but doesn't matter. Let's just go through them. Okay, so lipids. Okay, so we have this one. Okay, you should recognize them. Okay, when I show you pictures of these, you should recognize them. Okay, not the sugar. I'm not going to show you the sugar, but these one you should recognize them. So this one is triglyceride. Okay, also known as fat. And the components that makes up the fat include this thing right here. That is called glycerol. Okay, so one glycerol, and you have 3 of these fatty acids. 3 fatty acids. Okay, that's the building components of a fat molecule. Okay, triglyceride. What do we need fat for? They are useful for insulation, as in keeping organs warm. They are also good for long-term energy storage. Okay. We store short-term energy as glycogen, but then for long-term storage, we need to have the triglyceride of fat. Okay. Another kind of lipid that we talked about is this thing. Okay. There we go. This is the phospholipid. Phospholipid. Oops. Phospholipid. And we have the glycerol again right here. We have the 2 fatty acids. And there is also a new thing here that the triglyceride doesn't have. There is a phosphate group here. Phosphate group. Okay. So what do we need phospholipids for? Phospholipids, it turns out, it is the major component of cell membrane. Okay. Excuse me. The cell membrane is the membrane that surrounds the cell. And it has many things that makes it up, but the key ingredient is the phospholipid. Okay, phospholipid. Next, we also talked about steroid. That's another type of a lipid. And steroid has these, the major component of steroid is that it has these carbon rings. There are 4 carbon rings together. And then they have these like accessory that are stuck on them. Okay, and depending on which steroid you're talking about, these things might vary. The 4 rings are always going to be there, but these additional things, it might change. It might be different from one steroid to another. Collectively, we call these like functional groups. Functional groups. And what do we need steroid for? It is also a component of cell membrane. And we also need cholesterol to make hormones. Okay, they form hormones, such as the sex hormone, like testosterone and progesterone. Okay. That's pretty much it, guys. That's all the information that you have to know for the quiz. I mean, it's it's not a lot, I would say, but then it's not very little either. Okay, so you have to do your part and go through these things on your own and make sure you have good understanding of it. Yeah, that's it. Thank you for watching. If you have any questions, feel free to email me. Okay, good luck on your quiz and see you in class. Bye bye.